Welcome back, everyone, to session 2. We will have our next speaker presenting very soon. Our next speaker is Dr. Ala Abzane. He's a bioequivalence assessor in the Division of Bioequivalence 1, Office of Bioequivalence, Office of Generic Drugs at FDA. Thank you for presenting, Dr. Abzane.
Good morning, everybody. Thank you for the introduction. Today, I will talk about the expectation and common deficiency that we usually come across while testing in vitro release testing studies submitted and done for ophthalmic emulsion products. So before I start, just a disclaimer that this presentation reflects only my own views and should not be construed to represent FDA views or policies. So, the outline of my presentation, I'm going to talk briefly about the ophthalmic emulsion product and the role that IVRT play in the demonstration bioequivalence for ophthalmic emulsion product. And I will then move on and discuss the general expectation for submitting high quality IVRT studies and how a bioequivalent assessment of IVRT studies take place. Then I will summarize the most common deficiencies that we observe during the bioequivalent assessment of IVRT studies. And finally, I will provide suggestions and take-home messages for today's listener. As you know, ophthalmic emulsions are topical, locally acting formulation designed for ocular drug delivery. They are complex formulation mostly available as oil and water emulsion, where the active ingredient is dissolved in oil and the oil globule is dispersed in an aqueous phase containing water-soluble components such as surfactant, preservatives, among other ingredients. And therefore, the ophthalmic emulsion is considered complex equilibrium that occurs among different phases of the emulsion. And the amount of drug in the aqueous phase varies based on the equilibrium solubility of the drug and the partitioning coefficient. An IVRT study, as you have heard today, may be used to the part of the batteries and Vitro bioequivalence test to demonstrate that two products with similar formulation have comparable drug release rates. And you reflect the combined effect of physical and chemical properties of the active ingredient, the drug product formulation, as well as the manufacturing process on product and performance. As others described, the IVRT study doesn't need to mimic the in vivo condition performance or predict the therapeutic effect of the drug. Examples of the oil and water ophthalmic emulsion product, cyclosporine ophthalmic emulsion, difluprednate ophthalmic emulsion. These two have the product specific guidance, bioequivalent recommendation for these two products. They recommend that, among other things, acceptable comparative in vitro IVRT study of the active ingredients from the test and reference formulation. And the methodology used for the in vitro drug release testing should be able to discriminate the effect of process variability and the production of test formulations. So, in recent years, we have observed an increasing trend of ophthalmic emulsion and topical product submission containing IVRT studies. However, there are several challenges for IVRT studies submitted in support of the bioequivalence. This is because of the lack of compendial IVRT method. No specific USP Apparatus designed specifically to conduct IVRT study for ophthalmic emulsion products. And many of the deficiencies that we observe while assessing IVRT studies are related to incomplete submission because the applicant sometimes they do not provide report or information necessary to evaluate the suitability of the IVRT method for bioequivalent purposes. And this kind of deficiencies may be avoidable. And the bioequivalent deficiencies that we observe while assessing IVRT studies are often classified as major because it would require extensive assessment by the FDA. So in general, the bioequivalent, the IVRT study submission can be divided into 4 sections: IVRT method development, method validation, analytical method validation, and pivotal IVRT study. So it's preferred that separate reports are to be submitted for each of these sections. The applicant may provide step-by-step details of the test procedure so that the assessors are able to evaluate the appropriateness of the IVRT study conduct. And the applicant may also provide all the necessary data and calculations so that we are able to verify the result and make sure that it has been conducted appropriately. So regarding to what information to provide, which module to submit this information in the electronic common technical document, the applicant may provide and submit the bioequivalent summary tables in module 2.71. And in module 5.31, the applicant may submit the study report for each of the method, IVRT method development and IVRT method validation and analytical method validation, as well as separate report for the pivotal IVRT study. The applicant may also provide study protocols and standard operating procedures that were effective at the time of the IVRT study, and also prepare to have a complete numerical raw data and chromatogram for all samples that used during the IVRT study. And also, the applicant may submit all original, reinjected, repeated, and integrated analytical runs. And also, the applicant recommended to submit individual concentration data set used to calculate the drug release rate for the test and reference formulation in a test XPT format. By the way, this list is by no means to be complete list, and depending on the data submitted, additional information may be requested during the under bioequivalent assessment. Already, when talking about the bioequivalent assessment of IVRT study in a bit more detail, we look into the following information. And when it comes to IVRT study information, we like to applicant to provide complete information regarding the study number, study site, and the address, and details about the IVRT experiment date and sample analysis date. The complete information regarding the IVRT method description, the apparatus type used, the drug release medium, dose amount, and dosing application, and if the apparatus used a diffusion membrane, we like details about the membrane and the receptor solution, steering rate, among many other parameters. And come to analytical method description, we'd like to have information about the instruments used, the mobile field, buffer, diluent, chromatographic conditions, and also we'd like to have complete information about the test and reference lots used during the IVRT study method validation, as well as the pivotal study, including the lot number, manufacturing and expiration date, the potency and batch size. To understand why specific IVRT method parameters were selected and whether they are suitably sensitive and reproducible, For its purpose, we prefer the applicant provide detailed documentation on how the IVRT method was optimized and developed. And some of the parameters that we are looking to evaluate are sample loading volume, selection and sampling time point, information about the selection of the equipment and apparatus, the media receptor solution, the membrane temperature, agitation, rotation speed, pH sink condition, and surfactant type, among other parameters. And we'd like to have information about how the IVRT can capture the differences in the drug release resulting from differences in the formulation and manufacturing conditions. Overall, we would like the applicant to demonstrate that the method parameters selected for the IVRT study are appropriate and necessary. Additional information that we look for during the bioequivalent assessment IVRT study is information about the IVRT method validation as well as the analytical method validation. It should be noted that the information regarding IVRT method should be different from those used for the analytical method validation. And the information that's provided for the sample analytical method for you to quantify drug concentration in the IVRT receptor solution should be provided separately from those for IVRT method validation. And that validating the analytical method in itself doesn't demonstrate the validity of the IVRT method. So a separate report should be provided for each section. So for the IVRT method validation, we look into the method precision, repeatability, the intermediate precision, robustness. That would include intentional changes in the temperature, pH of the drug release medium, dose, and tearing rate, among other parameters. And we look on the membrane inertness and solution stability during the IVRT study, as well as the recovery and mass balance and the discriminator ability of the IVRT method. And when it comes to the analytical method, such as SPLC or UPLC, we look into this parameter for method validation, such as linearity, recovery, intra and inter-day precision, accuracy, selectivity, specificity, solution integrity, filter validation, solution stability. So during the bioequivalent assessment, the deficiencies that we observed during can be grouped and can be combined into the following group, which sometimes could increase the assessment time and the number of assessment cycles back and forth with the applicant. So typically, we see deficiencies that relate to insufficient details on the apparatus validation and experimental setup and method control. Also, we see missing information and insufficient data for the validation method parameters. Sometimes the applicant provides incomplete information regarding the discriminatory ability of the IVRT method. Sometimes we see insufficient information on the data analysis or data presentation, especially when the data is not what we are expected from the applicant. Because sometimes the applicants just tend to submit all the data, don't provide a clear graph or clear presentation, so we can properly evaluate the IVRT study. Also, the applicant may provide adequate documentation and organization submission of the IVRT report. Sometimes the IVRT information on the IVRT method development and validation are usually submitted in one single report. We prefer to have the individual report for each of the method development and validation. And sometimes the applicant do not submit information on the mass balance for the IVRT method due to diffusion membrane. It's not clear whether the drug is released from the formulation or because of the all globule transfer through the membrane. And sometimes the applicant forget to submit the raw data or such as XPK5, chromatograph, or SRPM. looking a bit more detailed about some of this missing information, like information of the IVRT apparatus used and the dose. Sometimes there are missing information, the membrane type, pore size, steering rate, and sometimes missing information on the detailed IVRT procedure, whether the membrane was pre-soaked or not, for how long, and if the pH rhythm was sequential during the duration of the IVRT study. And sometimes the applicant will provide inconsistent information throughout the submission. For example, the information that by equivalent summary table might be different from those provided in the detailed report. And sometimes incomplete data for studies involving multiple replicates is not clear of the number of replicates. Our summary statistics provide not individual data. And also missing protocol standard operating procedure, as well as lack of data and justification to support the optimization of the IVRT method, and missing information on how the apparatus was validated and qualified for demonstrator producibility and robustness of the IVRT method. And missing mass balance data, as I discussed previously, as well as missing data showing the disseminator ability of the IVRT method in terms of sensitivity, selectivity, and specificity. And explanation by the experimental data demonstrate the ability of the IVRT method to demonstrate the absence of process variability in the production test formulation. And when it comes to analytical method validation, sometimes applicants do not submit the SRP or protocol that were effective at the time with analytical method validation, or missing information on the IVRT sample processing prior to analysis. And sometimes the applicant does not submit information on the storage period and storage conditions for the IVRT sample for analysis. To summarize, take-home messages and my personal suggestion, the number of the submissions that contain IVRT studies has increased in the recent years. And as you have seen today, the IVRT study is a tool to ensure consistent performance and quality of the generic product and provide effective approach to monitor post-approval change, scale-up, lot-to-lot changes, and stability for ophthalmic emulsion. By equivalence related IVRT deficiencies are often classified as major because it requires extensive assessment. The lack of standardized method and tools for measuring in vitro drug release is a challenge for both the agency and industry. And many of these deficiencies that I have just presented may be avoided by providing detailed data and rationale to support the selection of the IVRT method, as well as providing complete and detailed information to allow for proper assessment of the appropriateness of the IVRT study conduct and the accuracy of the results provided. Finally, I would like to thank Amanda John, Dr. Amanda John, and Dr. Ophthalmic, and Dr. Josephine for their contribution and input in preparing this presentation, as well as the bioequivalent assistors and our colleagues at the Office of Research and Standards for their collaboration. Finally, thank you to the Division Bioequivalent Management for their support. Thank you very much.
Thank you, Dr. Abzane, for the presentation. Our next speaker is Dr. Hannah Pandal. She's a reviewer in the Division of Biopharmaceutics, Office of New Drug Products, Office of Pharmaceutical Quality at FDA. Thank you for presenting, Dr. Pandal.
Sorry, just a second. So can you hear me and see my slides?
Yes, we can, Kalpana.
Okay, thank you. Today I'm going to talk about common deficiencies and considerations on setting appropriate IVRT specifications for intravaginal rings from quality control perspective. So before I start, here is a disclaimer This presentation reflects my own views and should not be construed to represent FDA's views or policies. So in today's talk, I will cover about a brief introduction on vaginal rings, considerations on developing in vitro release test, and approaches for setting appropriate in vitro release acceptance criteria for these rings, common IVRT deficiencies in applications, and summary of my today's presentation. So vaginal rings are flexible polymer-based drug delivery systems for delivery of one or more drugs in the vagina. We are aware of different types of vaginal drug delivery systems, but this presentation is specific to the vaginal rings. So the ring typically allows for controlled release of the drug substance over an extended period of time, ranging from weeks to months. And vaginal rings releasing the drug up to a year has been approved. Depending on the specific vaginal ring design, it can be targeted to achieve a therapeutic effect for a local site of action with minimal systemic absorption or for the systemic absorption. So for vaginal rings to exert its effect, the API must be released from the rings. So the drug release profile of an IVR or intravaginal rings may have various phases of drug release: the burst effect or the early phase, transition or the middle phase, and a late phase. So this is a typical in vitro drug release profile of an intravaginal ring. So vertical axis is the rate of release expressed here as microgram per day. And the horizontal axis is in time, here in days, releasing the drug for 21 days. So this is the burst release phase that is characterized by an initial higher drug release rate that usually occurs within the first 24 to 48 hours of release testing. And thereafter, the drug release rate decreases, leading to a transition phase and to a steady state phase of release. So here are some general considerations for development of an in vitro release test for quality control purposes for vaginal rings. So there are two types of tests that can be developed: real-time drug release test and an accelerated drug release test. So in real-time drug release test, the in vitro drug release is evaluated using a release test period that reflects duration of use of the drugs. And this test provides mechanistic understanding of drug release kinetics. But when the real-time testing involves a long duration, applicants are encouraged to develop an accelerated drug release test for regulatory purposes. For this, a relationship between the accelerated and real-time in vitro drug release test should be established. And ideally, accelerated and real-time release should follow the same drug release mechanism. Therefore, evidence demonstrating a similar mechanism of drug release of two tests should be provided. And to evaluate the data, in vitro release data from biobatch and exhibit batches, including stability batches, using both real-time and accelerated tests should be provided. Also, efforts should be made in developing an in vitro release test that can release at least 80% of the label amount or when a plateau is reached. So whether it is a real-time or an accelerated test, IVRT should be discriminating to identify critical attributes that has potential to affect in vitro or in vivo release. So the selected method should differentiate the release profiles of the proposed target drug product and the test products that are intentionally manufactured with meaningful variations for the most relevant critical bioavailability attributes. So critical bioavailability attributes are formulation or process variables that are expected to critically impact the bioavailability of a drug product. Accelerated tests should show similar or superior discriminating ability compared to the real-time testing. And it is recommended to evaluate non-conforming batches that are out of specification under real-time test conditions, if available. So this figure shows the discriminating ability of an in vitro release test towards skin polymer thickness, that is the polymer thickness of the vaginal ring. Here the target batch has a skin polymer thickness of 0.102 millimeter, and the in vitro release profile for this batch is shown in gray color here. The in vitro release rate of aberrant formulations increased as the polymer thickness decreased from 0.088 millimeter and 0.075 millimeter. Similarly, the in vitro release rate decreased with an increase in the polymer thickness from the target thickness of 0.102 to 0.12 to 0.14 millimeter. So release rates at some time points were out of specification for the highest and lowest release profiles in this case, thus showing that the method is discriminating to the skin polymer thickness. So the proposed IVRT methods should be adequately validated. Data should demonstrate robustness, that it should remain unaffected by minor changes in testing parameters. Vaginal rings may be very sensitive to a change in temperature of the medium, and even a small change in temperature can give rise to high variability. Therefore, the temperature may need to be controlled within a narrow range if the temperature has significant effect on the drug release rate. Now, here are some considerations for setting appropriate drug release acceptance criteria for vaginal rings. From quality perspective, setting a clinically relevant drug product acceptance criteria is desired. Clinically relevant acceptance criteria are those that ensure consistent in vivo product performance of the formulation or the marketed product compared to that used in the pivotal BE study. So the goal of the in vitro release testing should be to develop drug product specifications that will ensure adequate performance of the future batches prepared within the limits of acceptance ranges. And another point to consider is that acceptance criteria should be set based on the release rate, that is amount per day. Here are some considerations for selecting time points for acceptance criteria. A minimum of 3 time points is recommended. Time points should cover the early, that is the burst effect, middle and late phases of the drug release profile. It is recommended that sufficient number of samples be taken to capture these phases. It is also important to clearly document the day the in vitro release corresponds to and provide an explanation if a different day is used for sampling than the set specification days. And the acceptance criteria should consist of ranges at each proposed sampling point, including the first and the last time points. So when there are no data linking in vitro drug release and in vivo performance, acceptance criteria should be set based on the data of the clinical batches of the proposed drug product. In vitro drug release data from 12 units of these batches should be used. In general, the drug release acceptance ranges is based on mean target value plus minus 10% at steady state, and wider acceptance criteria ranges may be acceptable if supported by a safe space approach. Here is an example of properly setting acceptance criteria for intravaginal rings. So this is the in vitro profile of a bio batch in blue and 2 exhibit batches. Again, vertical axis is the rate of release expressed here as microgram per day. And horizontal axis is in time here for days, and the drug is released for 21 days. This is the proposed acceptance criteria. So day 1 is not more than 360 microgram per day. Day 2 to 21, mean range from 96 to 144 microgram per day. And day 21, not less than 80 microgram per day. And in this case, our recommendation in such cases would be to establish in vitro daily release range for key stages. For example, day 1, which captures the burst release. And instead of mean range of release rates from many days, say here from 2 to 21 days, we recommend a single day in transition state. For example, in this case, it may be day 14, it may be day 5 or 4 or 6 in this transition phase, and a single day in steady state. So in this case, it may be day 14, around day 14 or 15, and day 21, the last day of the real-time use of the vaginal rings. So the final acceptance criteria should look something like this. Here, so the time points should cover the key stages of drug release, here like day 1, 6, 16, and 21. And the range should be set for all the time points, including the first and the last time point. Another way to set clinically relevant acceptance criteria for vaginal rings is via safe space approach. Safe space also provides regulatory flexibility in setting wider acceptance criteria, as I mentioned earlier. So safe space is established by defining the extremes of in vitro release rate within which drug product variants are bioequivalent to each other and to the target or reference formulation. So safe space can be established by various ways. One is based on the bracketing approach. Here, the ranges of acceptance criteria are established based on demonstration of bioequivalence between upper and lower proposed release rates. Another way to establish safe space is based on modeling and simulation. It can be done by physiologically based biopharmaceutics modeling, where limits are set based on using an appropriate approach, such as virtual BE studies, or via IVIVC modeling, where the limits are established based on a predictive and robust in vitro in vivo correlation. And in vitro release profiles and in vivo data generated during drug product development following formulation or process changes can be used to set the safe space if the data are available. So here is a scenario where safe space can be helpful. This is again in vitro release profile of a vaginal ring from 3 batches, bio batch and 2 exhibit batches. As can be seen here, bio batch and batch A have similar release profiles, while batch B is consistently releasing higher amount of drug compared to the bio batch. Now for vaginal rings, the release to some extent is controlled by a release controlling polymer. For reservoir type of vaginal rings, this drug-free polymer layer surrounds the drug substance containing core, and the thickness of this polymer layer is called the skin thickness. So usually there is a range set up for the skin thickness for the product. And in this case, the proposed range was 70 to 100 micrometer. And batch B was had the skin thickness of 70 micrometer, that was at the lower end of the limit, and was again releasing higher amount of the drug. Now, however, there are no data to evaluate if batch B will have comparable in vivo performance as bio batch. And this is a type of challenge we can face with vaginal rings. Now, if we have information that batch B will perform similar to biobatch, which can be done by conducting a BE study or by establishing a safe space by using IVIVC or physiologically based model, now this will be the opportunity to understand the product and make an informed decision to set clinically relevant specification. So in this case, if batch B is shown to be bioequivalent to the biobatch, then acceptance criteria range can be widened to include the higher release rate of this batch B. Now, here are some common deficiencies in the applications. So the method development report is not included. So a method development report with data demonstrating that the selected test is appropriate for the proposed drug product should be submitted. And methods should be product specific. And justification for this justification should be provided for selection of each in vitro release test parameters. Also, this point not mentioned here, but the method development report should also clearly specify the test conditions used at each development phase. And also the detailed validation of in vitro release method should be submitted. Another common deficiency is failure to demonstrate that the method is discriminating. So the first important thing is to identify or the identification of the critical bioavailability attributes. This is important. And then data should be obtained from target batch and batches with intentional meaningful changes. And appropriate statistical tests should be conducted comparing release profile of the changed versus the target batch. Another deficiency is data does not support the proposed acceptance criteria. So acceptance criteria should be based on the data from the proposed drug product, and time points should represent different phases of the drug release, and consider ranges rather than not less than or not more than for all time points, and submit complete in vitro release profile data that includes individual and mean values. So in summary, we recommend to develop an appropriate product-specific in vitro drug release test for vaginal rings. We also recommend developing an appropriate accelerated test along with the real-time drug release test. And data demonstrating that the selected method is appropriate for the proposed drug products should be submitted. And also data for demonstration of discriminating ability should be submitted. And we recommend setting clinically relevant acceptance criteria based on data obtained from batches used in clinical studies when in vitro in vivo link is not available, or setting the acceptance criteria by establishing a safe space by modeling and simulation or bracketing approach. So finally, I would like to acknowledge and thank my colleagues in the Division of Biopharmaceutics for all the support I received for this presentation. And I would also like to thank the organizers for giving me this opportunity. And lastly, I would like to thank you all for your time. Thank you.
Thank you for the presentation. Our next speaker is Dr. Diane Burgess. She's a distinguished professor of pharmaceutics at University of Connecticut and Pfizer Distinguished Chair of Pharmaceutical Technology. Thank you for presenting, Dr. Burgess.
Thank you for the invitation. Hello, everyone. So I'm going to talk on IVRT method development for LAI suspension products, and also the validation of that by using some in vivo preclinical models. So I'm going to have a few break slides throughout my talk, and they'll mostly be from Vermont. I spend some time in Vermont in the winters, in the spring, the fall, and in the summer. So this is from this summer. So the session today, as I said, will be mainly on the in vitro release testing method for the LAI parental suspensions. And our objectives were to develop in vitro release tests that would better correlate with in vivo release. In particular, we wanted to establish longer tests than what are currently available. We were going to compare some different testing methods and then hopefully develop the IVIVCs. So what are these LAI suspensions? So they're long-acting periods of weeks to several months. They're injectable IM sub-Q and also intraarticular. They can be for suspension or suspension. So for suspension, they're stored as powders and reconstituted prior to use. The ones that are suspensions can be in the form of oil or aqueous-based drug crystalline suspensions, and they're used in a number of chronic disease conditions, such as schizophrenia, diabetes, contraception, inflammation, pain control, cancers, etc. So the advantages are their extended drug release profiles. They can achieve the flip-flop pharmacokinetics, where the apparent half-life is controlled by the absorption rate constant, gives us predictable correlation between the dosage and the plasma levels. IVIVC should be possible. We have reduced peak trough plasma levels, consistent bioavailability, particularly good patient adherence, and reduced pill burden. For example, instead of, if it was 1 tablet a day, 365 tablets a year, we would only have 12 injections. If the injection was lasting for 1 month, let's say it was lasting for 3 months, then maybe only 4 injections. So definitely gives better patient adherence. So there are different kinds of these long-acting suspensions. One kind is the PLJ-based, particularly PLJ-based microspheres and in-situ-forming implants, such as Lupron Depro, Risperidone Consta. However, these are not what I'm going to be talking about today, but instead we're going to be talking about this slow drug dissolution from crystalline drug suspensions, such as the Deepa Sub-Q Provera. So there are several of these products on the market, such as the medroxyprogesterone acetate, which we're going to be focused on. But there are others, such as palperidone palmitate and aripiprazole. And so these are both as ready-to-use suspensions and powders for suspension. And you'll see that this from 2 to 4 weeks to about 3 months. So there are some recommended dissolution methods. However, all of these are fairly rapid. They're ranging from about 45 minutes to a couple of days. So our expectation was that these would not be so good to develop an IVIVC for something that is releasing from weeks to several months. So we set about developing some IVIVCs that would, some in vitro release testing methods that would be a little longer than a couple of days. So to do this, we focused on the Deepa Sub-Q Provera, and we looked at the composition of this, and it's got the API, obviously, some preservative, tonicity adjusting agents, suspending agents, surfactants, buffers, pH stabilizing agents, etc. So we looked to make formulations that were Q1, Q2 to this. But with some differences, so some things that we thought could affect the release could be the particle size as well as the size distribution. So to prepare some different, some formulations that would be slightly different, but we're still Q1, Q2, we did recrystallization of the API using an anti-solvent method. We also did some reduction in particle size by sonication. So we had 3 formulations initially. So the first one, we used the API just as received. The second one, we did the recrystallization. And the third one, we did some probe sonication to reduce the particle size. Sorry, that jumped there. So we did some characterization, such as the drug content, the uniformity, pH, drug solubility in suspension, also solid state characterization, such as PXRD, DSC, and obviously particle size distribution, and morphology. So you can see here of our 3 formulations together with the RLD. So the one that we did the recrystallization, we got the larger size. The blue one is as received. Red is the RLD. And then the one that we did sonication was much reduced in the particle size. That's the green one. This is another break slide. This is in Vermont about a month ago. And this is me on my bike, almost at the top of this particular mountain, which is had about 5 miles of climbing to get to that point. It's just starting to level out. So with the IVRT method development, we started with the apparatus 2 and the dialysis sacks because it's quite a popular method. We use a couple of different molecular weight cutoffs, the 100 kilodalton and the 1000 kilodalton. And we use the 300 ml mini vessels, we use PBS, we added some SDS and did it at 37. So for a sample loading inside these floatalizers, we used 100 microliters of a suspension and mixed that with 900 microliters of the 7.4 PBS. However, this method did not work out so well. We did an initial comparison with our formulation 1 and the RLD and found that we were not able to discriminate, even using the 1000 kilodalton, some very little discrimination, but you can see that the error bars are overlapping. And these were quite long tests. We believe that this was due to the very low sensitivity in differentiating the formulations was due to the violation of sink conditions inside dialysis sacks, which we reported for many number of years, going back probably even 30 years. I've been reporting on this problem with that you can have with the dialysis sacks method. So we also looked at IVR method development using USP Apparatus 2 with enhancer cells. And this at the side here, just showing you how we put these together. We've got the cell body, adjustable plate, membrane, gasket, and screw cap. One issue that we find with this is when you put the screw cap on, you can sort of wrinkle or even tear the membrane, and that can cause air bubbles or gaps or tears. And this can be an issue with the reproducibility in this method. But we did try this method nevertheless, and the conditions are listed here. And we found that we got very good discrimination between the RLD and F1, but we did see these relatively high error bars, which are quite typical of this method. We also developed an in-house method to kind of replace these adapters. And here we had a little kind of slide on top so that we could prevent any wrinkling or tearing. And one issue here was that we were making this base with Perspex, and it was quite difficult in our machine shop to machine it. So we're working on improvement of this. So with our in-house design method, we actually got better discrimination between the RLD and F1, again, going up to, as in the previous one, like at least 14 days and beyond. But we did get this relatively high error bar, which we believe was due to the manufacturing method with the that we are working on to improve that now. And finally, we used the apparatus 4 method with semi-solid adapters. You can see here our semi-solid adapters and how they're assembled. And we don't put a membrane on the top of these, but we do obviously have membrane up here at the top. We have 2 glass fiber filters up at the top to prevent sample going out of the flow-through cells. So with the semi-solid adapter method, this was a little bit faster. It was more like 6 days or a week. But we did get reasonably good discrimination, and we got very small error bars. So this was very reproducible method. And we also looked at the impact of flow rate on the release rate, and it really didn't have much of an effect. Also, again, showing the excellent reproducibility of this method. So comparing our three methods, we have the USP Apparatus 2 with the enhancer cells, the in-house designed one, and the USP Apparatus 4 with the semi-solid adapters. And we compared these for all our formulations, formulation 1, 2, 3, as well as the RLD. And you can see that formulation 3, which was small particle size, was the smallest in all the cases, sorry, the fastest. And then the slowest would be the one that was recrystallized. So we then went on to start our in vivo studies, and we actually prepared one additional formulation. So this was the F4 formulation, which was exactly the same as F1. So we didn't do anything to the particle size, but we used a different vendor for the PEG 3350. We used the Spectrum Chemical for F1, and we use BASF for F4. And we have previously reported that when you do use some different excipient sources, particularly for polymers, then this can affect the final product in many cases, and so can affect the in vitro and in vivo release. Sorry about that. So we did add this as an additional formulation with the rabbit studies. So for these 4 formulations and the RLD, we went on to do our rabbit studies, but we elected to use the Apparatus 2 with the enhancer cells and the Apparatus 4 method. And we didn't use our in-house designed one because we were, as I said, we were working on manufacturing that with more precision. So these are the in vitro data, just summarizing for and including this 4th formulation in both of these cases. So this is my next break slide, and this time I'm on my gravel bike in Vermont. And so the PK profiles for the LAI suspensions for the 4 formulations and the RLD is here. Looks like a big mess of data points here, but we did the in vivo deconvolution. And one of the things that was very surprising was that, F3 did not fall where it should have fell. It should have been, we thought, the fastest one, but in vivo, it turned out to be the slowest. So we did some investigation of this. And we found, in fact, that this one where we had reduced the particle size quite significantly, it had become unstable and was increasing and growing in size. When we studied it over a period of 10 days, it kind of started to stabilize around about day 5 or 6, and the particle size had significantly increased. So we believe that this is a reason why the F3 does not follow the expected rank order in vivo. In vitro, however, we're diluting it into a very large volume, whereas in vivo, it's going into the subcutaneous space. And so there's not much room for it to get diluted, and a lot of potential for it to aggregate. Another possibility in vivo is that it could be going into solution rapidly as well, and then recrystallizing out in that space. So we based on this instability, then we decided not to pursue with the F3 to develop our IVIVCs because of this instability. And we could have, of course, reformulated to add some additional surfactant or something that would aid in the stability of that, but then we wouldn't have been Q1, Q2. So to develop our IVIVCs for these LAI suspensions, so we tried with the data from the USP Apparatus 2 with the enhancer cells, and we used formulations F1, F2, and RLD, and then we tried to predict the formulation 4, and we used the double Weibull method. We had, and here we're doing IVIVC just using F2 and the RLD and predicting F1. And you can see that in our prediction error for the AUC was really good. It was less than 10%, but for the Cmax, it wasn't. It was above that. So this was not a good IVIVC. And then again, we used F2, F4 and the RLD, and we tried to predict F1. And again, with the AUC, the prediction error was really strong, but with the Cmax, it wasn't. So again, this was not a good IVIVC. We then worked with the data we had from the USP Apparatus 4. We used the formulation F1, F2, F4, and the RLD. And we used the models, the Weeble, when we tried with 4 formulations, and the Hill when we tried with 3 formulations. And with the Hill, with F1 and the RLD, we got very good prediction of F4, with both the prediction error for the AUC less than 10%. Also, the prediction error for the Cmax was less than 10%. And we also did prediction of F4 using F1, F2, and the RLD. And again, our prediction errors in both cases was less than 10%. And this was using F1, F2, and the RLD, and using the Weeble method. We also looked at IVIVC with F2, F4, and the RLD, and F1, F4, and the RLD. And so we used this one here to predict F1 and this one to predict F2. And we got, again, very good prediction with our data overlapping for most of the data points with the USP Apparatus 4 in vitro release. So I think the key with that one was that with the Apparatus 4, we had the very small error bars. And so we were getting this really good predictions. So to summarize the data, we developed compendial methods with minor modifications for the LAI suspensions. We showed good discriminability for our Q1, Q2 equivalent LAI suspensions. We got the best reproducibility with Apparatus 4, but with Apparatus 2, we got a little bit higher margin in the discriminability. However, the Apparatus 4 method did turn out to be able to develop the IVIVCs successfully. And we're also now working to increase our understanding of the IVIVCs for LAI suspensions by using other modeling such as PK. And with that, I'd really like to thank Dr. Chen-Ying Bao from my research lab. This is about a month ago when we were in Vermont together and we're climbing up Stratton Mountain. And you can see some beautiful view of the Green Mountains in the background. And just up here is actually Killington Peak looking from Stratton. And this is probably the Okemo over here. And that last picture was with her 4-year-old son, Aaron, and this is with her baby, who's like 7-8 months old now. And Mason is his name. And then just to acknowledge the rest of our research group here and the support from the US FDA and our collaborators at FDA, Yan Wang and Yuan Zhou. This is pre-pandemic, we're not having our masks and we're all together, but this is a recent graduation from one of my students, Gautam here, just graduated. And so, of course, we're all duly wearing our masks. So with that, thank you all for your attention.